Well, thank you everybody for tuning in. My name is Maureen Ascona. I am currently a third-year graduate student in the Legal and Brembilla labs. And today I want to discuss a little bit about my project, Astroglial TNFR2 signaling and synaptic and cognitive function after TBI, and its role in astroglial D-Serine release. So I just want to make a quick note before I get started that I am fairly early into my research career. So, a lot of these concepts are going to be more conceptual based. So if you really have any questions, feel free to stop me at any time. With that being said, I'm going to go ahead and just get started. For now, I'm going to have a brief introduction on traumatic brain injury with some explanation on some of the background of why we've been doing this. Then I'm going to expand on my hypothesis, my working model, and then I'm going to conclude with my aim 1 and aim 2 and the plans that I want to achieve for this. So with that being said, I will begin. So traumatic brain injury is really this very prevalent issue where approximately 2.87 million people are affected each year by traumatic brain injury. Of those, approximately 65% of moderate to severe and 15% of mild traumatic brain injury patients experience some form of long-term cognitive deficits. To highlight a little more on the prevalence of cognitive deficits in TBI patients, a study was done with Ponsford in 2014 that highlighted memory issues and difficulty concentrating and planning. These sort of ideas focus more on cognition, on how patients who experience traumatic brain injury are sort of experiencing these complications. And what was interesting about this study is that 2, 5, and 10 years after injury, regardless of level of severity, there seemed to be no amelioration in these types of cognitive complications over time. So just to highlight the importance of this cognitive impairment after injury, which is why I've decided to focus my studies on the cognitive deficits that arise from a result of traumatic brain injury. So to expand a little more on TBI, TBI is really known to have 2 phases. The first is a primary injury phase. This can be really defined as the mechanical forces that impact the brain immediately after injury, and can be characterized with damage to blood vessels, compromising of the blood-brain barrier, things like axonal shearing and tissue damage, which often results in neuronal cell death, depending on the location of the injury, as well as a more secondary phase. This phase is more defined as the physiological and molecular alterations that occur as a result of primary injury and are more focused on ischemia, edema, alterations in neurotransmitter release, changes in free radicals from oxidative stress and mitochondrial damage, as well as increased reactivity that results in gliosis. So we often affiliate these secondary effects with synaptic impairment, which is often affiliated with cognitive impairment. So this is why I've decided to focus a lot of my studies on, since there are really no treatments that focus on the secondary complications after TBI. So to expand a little bit more on what is considered a standard synapse, I've got just here a quick demonstration of a typical pre- and postsynaptic cell and how astrocytes are able to modulate this through a very important tripartite synapse. Here, the astrocytes are able to both maintain homeostasis and allow for the modulation of synaptic activity through various means, either through buffering of potassium or glutamate and the synaptic clef, to a release of gliotransmitters to alter synaptic activation, as well as aiding in formation and elimination of synapses, and primarily modulation of synaptic function through TNF signaling. So to expand a little bit more on TNF signaling in astrocytes, even though we understand TNF, tumor necrosis factor, to be this cytokine that's implemented in inflammation, it actually plays a significant role in synaptic function. It's able to do this through the alterations of synaptic scaling, by strengthening glutamatergic synaptic activity through AMPA receptors, as well as by modulating synaptic plasticity. So this cytokine is really able to function through two primary receptors. So we have a TNFR1 receptors. These respond primarily through binding of soluble TNF to TNFR1. This mainly has been more affiliated with a pro-inflammatory state and usually results in detrimental effects due to downstream cell death or inflammatory mechanisms, while TNFR2 is primarily activated through transmembrane TNF, which binds to TNFR2 receptors with more of an anti-inflammatory and neuroprotective effect. Downstream TNFR2 signaling has been shown to be non-canonical NF-kappa B signaling, as well as PI3 kinase or PI3K signaling. And what's really interesting about TNFR2 is that its downstream signaling pathways have been shown to alter glucose metabolism. So it's been shown to do this in the PI3K pathway through alterations in glycogen synthesis. ATP is able to activate glycogen synthase by inhibiting its inhibitor, resulting in an increase in glycogen synthesis. As well as in the non-canonical NF-kappa B, it's been shown to alter glucose through increases in glucose utilization via glycolysis through the increase of proteins and genes involved in the pentose phosphate pathway, as well as upregulation of this pyruvate kinase, which is involved in the final step of converting PEP to pyruvate. This this glucose intermediate that is able to be either converted to pyruvate to be able to enter the citric acid cycle or to be made to lactate, which can be used in various means. And to bring a little bit more emphasis on the important role that astrocytes play in glucose metabolism, I just want to show this figure to really say that astrocytes are able to increase their production of lactate and glycogen in order to shuttle to neurons whenever neurons are in a state of high metabolic demand. So I'm not really going to go into the detail of any of these things, but it's really just to emphasize that neurons and astrocytes really show a tight metabolic coupling and have very strong glucose metabolic connections. Now, to expand a little bit more on another important player that is involved in this glucose story, I want to talk a little bit about D-Serine. So D-Serine is derived from L-serine, which is the precursor. And L-serine is derived from 3-phosphoglycerate. It's involved in one of the steps in the glycolysis pathway. So this 3-phosphoglycerate is able to bind to PHGDH, which will then allow it to go into the L-serine biosynthesis pathway. From here, L-serine is synthesized in astrocytes. It is then transferred over to neurons, in which the enzyme serine racemase can then convert L to D-serine, which can then be released to allow for normal cognitive function. So the normal cognitive function that D-serine primarily lies with is it's the primary coagonist for NMDA receptors in the hippocampus. So what it does is that it binds to this glycine modulatory site in the N2, N1 subunit of the NMDA receptors and allows for basic NMDA functions, which are usually coupled with synaptic plasticity and LTP. Another interesting point to address is that D-serine is really highly expressed in neurons. So this is to mainly say that D-serine is mainly found in neurons where it can be released to cause normal function. It's approximately 90% of neurons experience or have D-serine compared to approximately 10% on astrocytes. And just something interesting that was uncovered in the Libo lab was that we found that after traumatic brain injury, there seems to be a switch in the release of D-serine from neurons to astrocytes, where increases in serine racemase are able to convert this L-serine that's in astrocytes to D-serine for it to be released. Studies from the Libo lab have also demonstrated that actually the release of D-serine from astrocytes has been shown to lead to impaired cognitive function. And I'll expand a little more on that. So in order to have a model for us to demonstrate traumatic brain injury, we've decided to use a controlled cortical impact model. In this model, an animal receives a craniotomy at a specific size, and then a pressurized piston will go ahead and fire at a rate of 4 meters per second in a depth of 0.5 millimeters. And what will happen is that there will be an impact on the cortical structures, but ultimately the hippocampus will be saved. And what we'll see is there's going to be, if we pay attention to the number of new and positive cells in the CA1, CA3, and dentate gyrus, all of the different regions of the hippocampus, we find that there's no alterations in the amount of new and positive cells, indicating that there is no neuronal cell death that occurs. But if we look at the number of synapses at the CA1, there is a significant decrease in the amount of synapses. So this is important for us because it allows for damage to the hippocampal synapses without altering cell death. This is important because we are primarily interested in the effects of synaptic impairment and how it results in cognitive dysfunction. So to expand a little more on the role of D-Serine and how it may be altering cognitive function, we have an LTP study that was done in the lab previously. This is a little bit complicated, so I'll walk you through it. Here we have a serine racemase flox animal. This is a control animal in sham conditions, which they received no injury, and a CCI condition. Here we see that after high frequency stimulation, the sham is able to successfully learn or have successful LTP, while the control animal is impaired in its LTP. I'm sorry, the CCI animal is impaired in its LTP. If we look at our transgenic serine racemase animal that's been knocked out, specifically in astrocytes, in sham conditions, there doesn't seem to be any alterations in the ability for the animal to experience high frequency LTP from high frequency stimulation. But if we look at our knockout animal in a CCI condition, we find that there's actually a rescue. This is indicating that if we actively remove the ability for astrocytes to convert L to D-serine for it to be released, then we ultimately improve the ability to achieve LTP. This is to say that the release of D-serine specifically from astrocytes is resulting in impairment. If we also take a look at what happens when we alter TNFR2 expression in astrocytes, here we have an animal that has been knocked out of TNFR2, specifically in astrocytes, and then we observe what happens in LTP. So after high frequency stimulation, we notice that the animal that has received the removal of TNFR2 has impaired LTP after, and this is just in a naive condition. This is just to say that this TNFR2 is necessary for basic synaptic function and basic function for LTP. So to just bring this together, really, D-serine has been shown to be, once it's shifted to astrocyte release, impairing in cognitive function, at least in the LTP level, and TNFR2 is necessary in order to have proper LTP expression. Because these both have been shown to be linked in some way or another to glucose, where TNFR2 would be upstream of these, and D-serine would be more of a downstream product. I'm starting to think that these two might be linked. So to have some conclusions from a previous descriptions here, we've really shown that astroglial D-serine can be detrimental after injury when released, and that astroglial TNFR2 ablation really kind of recapitulates a lot of the things that we're seeing in the TBI conditions if we take a look at the LTP data. But what's mostly interesting is that the mechanisms that are regulating this astroglial D-serine synthesis are largely unknown. So because of these links that have been made and the way that removing TNFR2 works in the same way of releasing astroglial TNFR2, I've really come up with this overarching hypothesis that astroglial TNFR2 signaling plays a protective role on synaptic and cognitive function after traumatic brain injury by modulating glucose metabolism and thus D-Serine biosynthesis. So I'm going to expand a little on how I believe this is happening through a working model. So in this model, here we have in physiological conditions, TNFR2 expression that is able to activate downstream non-canonical NF-kappa B signaling, as well as PI3K signaling, which works to, in its own pathways, alter glucose metabolism and utilize intercellular glucose, as well as the standard L-serine biosynthesis pathway, where L-serine is made and then is able to be shuttled to neurons, where it can then be used for basic biological functions by conversion of D-serine. Then we have, in a traumatic brain injury conditions, what I believe is occurring is in this condition, while TNFR2 is activated, it's not upregulated enough to combat a lot of the sequestering that is occurring from intracellular glucose into the L-serine biosynthesis pathway. So preliminary data has shown that PHGDH and PSAT1, both enzymes involved in the L-serine biosynthesis pathway, are upregulated after injury, as well as serine racemase. So it's really this demand increase in the sequestering of this L-serine biosynthesis pathway that's really driving intracellular glucose to produce increased L-serine that can then be converted to D-serine and released from astrocytes to result in excitotoxicity. What I propose is a gain of function for TNFR2 in traumatic brain injury. So if we increase TNFR2 to increase a non-canonical NF-kappa B and phospho-AKT signaling, what we'll find is that there's going to be an increase in glycogen synthesis, as well as increases towards the pentose phosphate pathway and increases in ATP and lactate production that will effectively deplete intracellular glucose so that there is less glucose available to be shuttled into the L-serine biosynthesis pathway. What's interesting is that PHGDH has a very low affinity for 3-phosphoglycerate. So if you deplete this intracellular pool, it should be fairly effective at minimizing the amount of serine that can be synthesized. If you minimize that L-serine, it will minimize the amount of D-serine that can be converted that would ultimately result in excitotoxicity. And I also propose a loss of function for TNFR2 model in traumatic brain injury, where if we effectively remove TNFR2 and its downstream signaling mechanisms, there will be no alternative pathways for intercellular. Well, I mean, surely there are other ways for glucose to be utilized, but there will be a minimized way for glucose to be used up in the cell, which would allow for more intracellular glucose to go into the L-serine biosynthesis pathway and ultimately increase the production of L and D-serine to be released to cause toxicity. So I will now move on to the aims of which I want to use to prove that this hypothesis is actually what is occurring in a system. So my aim one really will be to determine the role of astroglial TNFR2 and synaptic and cognitive function after CCI injury. And my aim two will be to determine whether astroglial TNFR2 regulates D-serine synthesis after traumatic brain injury by modulating glucose metabolism. So I'm going to expand a little bit more on the models for the animal models that I'm using. Here I have my conditional TNFR2 knockout animals in astrocytes. So what we have here is a Cre recombinase specifically bound to a GFAP astrocyte-specific promoter. This is bred with an animal that has LOXP sites in our TNFR2 target gene. This animal will then be bred to become a GFAP Cre TNFR2 flox that, when in the presence of tamoxifen, will bind to the estrogen receptor and allow for the Cre recombinase to translocate into the nucleus, where it can effectively flox out the TNFR2. Our other model is our overexpression model. This is an inducible TET-off system where we have a tetracycline transactivator bound to a GFAP astrocyte-specific promoter that in the presence of doxycycline will become inactivated. When doxycycline is removed, which we introduce it in the chow. So when it is removed, this tetracycline transactivator will activate and can bind to a tetracycline response element to allow for the successful overexpression of TNFR2. So I will use these animals in a series of experiments throughout my AIM-1, and here is really just an experimental timeline of what I want to do with both of these animals. Really, for these two, I want to look at in time points 1, 3, and 7 days post-injury, some flow experiments as well as HPLC and IHC analysis. I also want to look at behavior in terms of Morris water maze and fear conditioning and novel object, and spine analysis as well as LTP experiments. So I'm going to now expand a little more on each one of these experiments. Beginning with behavior. So the reason I'm wanting to do behavior and the question that I wish to answer here is if altering astroglial TNFR2 either through overexpression or removal is going to have some effect on cognitive function after traumatic brain injury. So I first want to look at fear conditioning experiments. In these experiments, we first add an animal into a box and allow them to habituate. On day 2, we then introduce a tone and follow it with a shock. And on the final day, they are experiencing both a context and a cue experiment where the context will be similar to the habituation, except the amount or percentage of freezing as a factor of learning will be evaluated. And then a cue where the environment will change and the tone will be reintroduced to see the animal if they are able to affiliate the tone with the shock. Next, I want to do some novel object experiments. An animal will be placed in a box. The next day, the animal will be given 2 immediate similar objects, and they will be on the 3rd day introduced to a novel object as well as the same object from before, and we are able to assess the amount of time that the animal spends exploring the novel object. And the final experiment will be a Morris water maze experiment where we place the animal in different starting points and allow them to seek for a hidden platform that is somewhere in the maze. So I'm currently working on the novel object experiments right now. But for the fear conditioning and Morris water maze, there are some new systems that are being set up, and I'm very excited to do these experiments. So more to come soon. My anticipated outcome will be that if you have removal of TNFR2 in injury conditions, you're going to see impaired learning and memory. And what I expect is to see impairment that is further than what is seen in the control injury animals. And for my overexpressor animals, with injury, I expect to see improved learning and memory similar to what I would see in a control sham animal. So if we end up seeing the behavioral deficits that we expect to see, it would be interesting to see, does this impairment transfer over and this recovery that we expect to see, does it transfer over to electrophysiological alterations? This is why we want to see some electrophysiology experiments at 7 days post-injury. We want to initiate with LTP experiments where if we introduce high-frequency stimulation, will we see changes in the field excitatory postsynaptic potentials? We also want to do a paired pulse facilitation to test the presynaptic release probability alterations, as well as input-output to measure the efficacy of neurotransmitter release. So what I anticipate from these experiments will be that if we remove TNFR2 from these mice in injury conditions, we're going to have this decrease or impaired LTP and paired pulse facilitation as well as decreased input-output curves. And for our overexpressor models, in injury conditions, I would expect to see this increased LTP and paired pulse facilitation with increased IO curves. We are currently in the process of setting this up with a collaborator in the Lois Pope building. So I expect to be able to start this fairly soon. And I'm very excited to learn this new technique, as I've never done electrophysiology before. So very excited. Next, we're thinking, if we see alterations in electrophysiology and behavior, how might this be impacting the actual physiological morphology of the actual cells? So, which is why we've decided to do a dendritic spine analysis to see the effects of TNFR2, either overexpression or removal in astrocytes, and how that might be affecting the morphology of spines. So in this experiment, essentially what we do is we pick 3 different cells from an animal. And in those cells, we pick 3 dendritic arbors from each of those cells, and then we analyze the spines. So depending on the width and length of the spines, it will affect the morphology. And this is what we're going to be analyzing. So mushroom spines, which are a little bit wider, are considered more mature spines, while longer, thinner spines might be considered more immature or degenerating. And really, what I anticipate to see from these studies is that if I knock out TNFR2 in injury conditions, I'm going to see decreases in the spine density, as well as decreases in mature mushroom spines and increased immature and degenerating spines. And for my overexpressor animals, in injury conditions, I expect to see really no significant changes, as it should be ameliorating the injury or the synaptic impairments that are seen. So here actually are some images from the spines that I've already collected. I'm actually finished capturing the images, and I'm really in the process of doing this analysis. So this data is soon to come, and I'm very excited for the results that are going to come out from these studies. So now that we've really thought about how we're going to be observing these on different types of structural levels, we're also wanting to see how this might be affecting glial reactivity, which is why we've decided to do some IHC studies. This was something that I was able to achieve recently, where we have an astrocyte TNFR2 knockout. So this is specifically what we're looking at. And here we have a GFAP stain. So this is looking at astrocytes at 7 days post-injury. So if you take a look to your left, here we have our control animal and our knockout animal and sham and CCI conditions. In control sham conditions, we have basal GFAP expression. But what's interesting is that when we knock out TNFR2, in a sham condition, we see increased GFAP expression. This is interesting because if you look to your right, these are some studies that were done by our previous postdoc in the Brambilla lab, Placido, who was able to demonstrate that in TNFR2 versus or TNFR2 flox controls versus knockouts, he showed increases in GFAP expression, indicating increased gliosis in a naive condition, and my sham seems to recapitulate this, which is a good sign. For our CCI in our TNFR2 control versus our knockout, they're both increased in their reactivity compared to that of a sham. But if I was to truly determine if the reactivity is stronger in a knockout compared to a CCI, really a more quantitative evaluation would be more indicative of exactly what's going on. This is an N of 1, so more studies need to be done in order to truly make a conclusion, but so far looking fairly promising. If we also look at some of the expression that was done in IBA1, this is in the same animals, just in a different channel, again being TNFR2 knockouts in a 7-day post time point. Similar results occur where in our control sham, there's this basal IBA1 expression that is increased in our in our knockout animal, which is recapitulated from the studies that were done by Placido, where he has increased reactivity in the knockout in sham conditions, or in his case, naive conditions. And from our control and knockout in CCI conditions, there is this increased reactivity, although again, to determine more quantitative measures would be taken. So besides just looking at glial reactivity, we want to also use IHC experiments to look at in 1, 3, and 7-day time points. Again, not only the glial reactivity alterations, but also changes in serine racemase location after injury. So some previous studies done in the Liebel lab demonstrated that serine racemase expression is very strong in in the pyramidal cell layer in sham or non-injury conditions. And that once you introduce an injury, it actually shifts the expression of serine racemase to glial cells. So I would be interested to see how alterations in Astroglial TNFR2 are able to change, or if they are able to change the serine racemase expression after injury. So we're actually working with a collaborator on these serine racemase IHC experiments. This is Dr. Derek Balu from the Harvard School of Medicine. He's a leading expert in serine racemase, and we've actually already sent him some samples and are eagerly awaiting to see if there are any changes in serine racemase expression in both our knockout and overexpressor animals. So my anticipated outcome here is really to see how if knocking out TNFR2 in injury conditions leads to this increased gliosis as well as increased serine racemase glial expression. And for our overexpressor animals in injury conditions, will this effectively decrease gliosis and lead to decreases in serine racemase glial expression? So links have already been made from previous experiments that I've done on really TNFR2 and serine racemase. I have here a Western blot that was done that demonstrated that by effectively knocking out TNFR2 and astrocytes, you see changes in the protein expression of serine racemase. Now, it might seem a little peculiar that there's a decrease as you would expect an increase, which is more affiliated with a detrimental outcome due to the fact that this is a knockout. But if you recall from the beginning of my talk, 90% of neurons are really expressing this serine racemase and this D-Serine expression. So what we believe is occurring is that there's decreases in the serine racemase that we're seeing over here in neurons, as this is whole hippocampal lysate. So because we are in need of an astrocyte-specific analysis of what is occurring in serine racemase, we want to do some studies to follow up doing flow. So flow would help us determine if alterations of this astroglial TNFR2 not only would change the astroglial reactivity or glial reactivity in general, which is already being looked at in IHC analysis, as well as how it's affecting serine racemase expression. So we want to do flow analysis at 1, 3, and 7-day time points, where we can determine if this astroglial alteration in TNFR2 does result in changes in glial cell numbers in astrocytes and microglia, in turn reflecting changes in reactivity, and does it alter the serine racemase found in astrocytes. We also want to see if it alters phosphoate AT expression, but this is something that we want to look at in earlier time points. Some preliminary data has already demonstrated that in microglia, there are already increases in the amount of microglia found in the TNFR2 knockout in injury conditions, although this is very preliminary data and much more needs to be done to determine this. But this is at a 7-day time point as well. And really the anticipated outcomes that we expect from this experiment are to show that in knockouts, in injury conditions, there should be elevations in serine racemase expressions, as well as the glial cell numbers indicating increased reactivity, as well as decreased expression of phospho-AKT. And this is something that we would like to determine on an earlier basis. And for our overexpressor animals, in injury conditions, we want to see decreased serine racemase expression, as well as decreased glial cell numbers indicating of decreased reactive environment, as well as increased phospho-AKT expression. And the final experiment that I want to explore in my AIM-1 is going to be an analysis of how this alteration in astroglial TNFR2 might be affecting hippocampal neurotransmitters. And I really want to look at this through high performance liquid chromatography or HPLC experiments at 1, 3, and 7-day time points. I really want to see if this is altering all the levels of L and D-Serine. So we're actually going to be looking at this through a collaborator, Dr. Herman Wallisker from the Israel Institute of Technology. So Dr. Wallisker is an expert in HPLC experiments. He's also a leader in L and D-Serine research, and we are very excited to be working with him. I have not yet begun to do the collection for these experiments, but I'm very excited for what's to come. And really the anticipated outcome that we have for these experiments is that in a knockout injury animal, we expect to see increased D-Serine production, right, as we remove TNFR2 when intracellular glucose can't be reallocated. And then in an overexpression model with injury conditions, we expect to see this decrease in L and D-serine production because this glucose is being utilized to other locations. So really that wraps up my aim 1 and what I wish to achieve. So now I'm going to expand more on my aim 2. So in my aim 2, I really want to determine whether Astroglial TNFR2 regulates D-serine synthesis after traumatic brain injury by modulating glucose metabolism. And I mainly want to achieve this using an in vitro approach. So here I'm going to expand a little bit more on the timeline for my in vitro experiments. So I want to collect wild type pups at P2 to P5 time points, dissect out the hippocampus, and sort out the astrocytes and culture them. So I want to culture my astrocytes for 7 days. The reason I have chosen 7 days is because previous research from our labs has shown that at 7 days, these astrocytes take on a reactive phenotype and start production of D-serine, which would be a great model for our injury conditions. So at 7 days, we want to introduce a TNFR2 activating antibody, and we want to test to see if it's actually stimulating non-canonical NF-kappa B and AKT pathways. We want to then assess how this might be altering gene expression, as well as different metabolic products like glycogen, ATP, and lactate, and how it's altering L and D-serine levels, as well as how it might be affecting other proteins that are significant players in the story. And then we want to come back and introduce an inhibitor of either NIC or AKT, and then see how this might be affecting the results, the outcomes that we see from these previous results. So to introduce some of the inhibitors that we want to use to inhibit these TNFR2 downstream signaling pathways, we have a NIC small molecule inhibitor. This is inhibiting for the non-canonical NF-kappa B pathway. This NIC is a selective NIC inhibitor, which inhibits the NIC catalyzed hydrolysis of ATP to ADP. And studies have already tested it in vitro, in HeLa cells, in vitro and in vivo with its respective concentrations. And here we have an MK2206. This is an inhibitor of AKT for the PI3K signaling pathway. And this would be an allosteric inhibitor of AKT that prevents translocation to the membrane, which has been tested in cancer cells. And here are its respective in vivo and in vitro concentrations, which I will test through concentration experiments. So because this is the first time that I've done cell culture work, I first wanted to determine if I can make pure astrocyte cultures. So here is an image of some hippocampal cell cultures that I was able to do. I did both hippocampal and cortical astrocyte cultures. And what we have here is a demonstration of the percentage of overlap that I had with DAPI positive cells and GFAP positive cells, where I experienced somewhere around a 97% overlap, thereby indicating that I can successfully create pure astrocyte cultures. Next, what I wanted to do in these cultures is to introduce an activating antibody and then see if this antibody is indeed activating some of the downstream signaling pathways that I expected to. So we began with the PI3K signaling pathway by testing out it with Western blot phospho-AKT to AKT expression. Oh, sorry about this little mistake here. So if we look at our charts here, this is for a hippocampal culture, and this is for a cortical astrocyte culture. And what we find is I was able to collect the lysate after 5 minutes, 10 minutes, and 4 hours in this cortical culture, and only at 10 minutes due to minimized culture size for the hippocampals, just because there is a lot less tissue available. But regardless, if we introduce this activating antibody, we do see increases in phospho-AKT to AKT expression in both the 10-minute time points and the 5 and 10-minute time points in the cortex, which is not surprising because this is transcriptional factors that usually occur within minutes. This actually is not complete. This is one biological sample that was run in triplicate, but we still do need 2 biological samples in order to complete it. I have collected both of those biological samples and their lysate and their protein concentrations have been run. I just need to complete the Western blot to finalize these studies. I also want to then look at a P100, P52 marker to confirm a non-canonical NF-kappa B activation in these. So after we confirm that our activating antibody is successful in activating their downstream signaling pathways, we want to see how this is altering L and D-Serine levels and protein expression of some significant players. So we have here the question of, does activation of TNFR2 in reactive astrocyte cultures result in the alteration of L and D-Serine levels? And does it also result in alterations in TNFR2 and TNFR1 receptors, as well as serine racemase expression, and this PKM2, this pyruvate kinase? So our anticipated outcome is that TNFR2 activation decreases L and D-Serine levels, right? As we turn on TNFR2, it should be diverting glucose away from into other places where glucose can be utilized. And for the TNFR2 activation, it would result in decreases in serine racemase and increased protein kinase, pyruvate kinase expression. And we want to see if knocking out this non-canonical NF-kappa B and this PI3K pathway, does blocking this TNFR2 activation in a reactive astrocyte culture reverse the alterations seen in L and D-Serine levels? And does it reverse the alterations that could possibly arise from this TNFR2 receptor, serine racemase, and this pyruvate kinase protein expression? And the anticipated outcome is yes, we hope that it does reverse it. This would address the link that we believe is occurring. And if we see that this TNFR2 is in fact altering our final product, which is L and D-Serine levels, we want to see, is it doing so through glucose metabolism? So does this activation of TNFR2 in a reactive astrocyte culture result in alterations in glucose metabolism through changes in glycogen synthesis, as well as changes in ATP and lactate production? So we anticipate that this activation of this TNFR2 through the non-canonical NF-kappa B and the PI3K will have increase in glycogen synthesis, increase in lactate, and increase in ATP production. And if we knock out either the non-canonical NF-kappa B pathway or the AKT pathway, or which is also the PI3K pathway, does blocking this TNFR2 signaling result in a reactive astrocyte culture reverse the alterations that we see in glucose metabolism? And the hope that we find is that yes, right, thereby connecting the link that TNFR2 is modulating D-Serine effectively through glucose metabolism. The final study that I want to do to really place everything together is to show if this activation or this inhibition of TNFR2 downstream signaling will regulate L and D-Serine levels and alter cognitive function in an in vivo setting. So if I introduce these inhibitors that I had in vitro, in vivo, does it result in synaptic and cognitive dysfunction that would be similar to what is observed in our TNFR2 knockout in vivo after TBI? So I want to explore this with a fear conditioning experiment, as well as dendritic spine analysis and HPLC to assess alterations in DNL serine levels. And just a general question that we could explore as we're working through this is the synaptic and cognitive function more like the impairments that we would see or the alterations that we would see in synaptic and cognitive function more dependent on the non-canonical NF-kappa B pathway, or is it on the PI3K signaling pathway by just merely inhibiting either one of these downstream signaling pathways or the other? And our anticipated outcome is that inhibition of either of these two will increase L and D-Serine production. It'll also decrease the total spine density and mature mushroom spines, as well as increase mature immature and degenerative thin spines. And finally, that it's going to cause impairments in learning. So with that being said, that is really the end of my talk. I really just want to really bring some emphasis on the significance of these studies and why I'm doing them. So I really want to expand on the body of knowledge of TNFR2 signaling in the CNS. There's very minimal work that's really been done on TNFR2. And most of it is in cancer and diabetes. So I really just want to expand a lot more TNFR2 research because the field really dominates in TNFR1 at the moment, as well as to just discover a novel mechanism that explains what these D-Serine alterations after injury that ultimately result in cognitive dysfunction. As we've seen, it's such a prevalent issue that it'd be really interesting. Cognitive dysfunction is such a prevalent issue in TBI that it'd be really interesting to see if this is why these mechanisms are altered. And really to uncover a therapeutic strategy to promote cognitive recovery after injury, as currently there are no treatments for the secondary effects of or the secondary phase of traumatic brain injury. So it'd be great to really start focusing on a different target to promote improved recovery after injury. And with that, I really just want to thank both of my labs, both of my mentors, both Dan and Roberta, or Dr. Liebel and Dr. Brambilla. They've just been excellent working with me throughout this entire process, as well as the Placido, the previous postdoc in the Brambilla lab, and Madeline in the Liebel lab, and all of my wonderful coworkers whom I couldn't possibly have done this without, Aretha, Brianna, Esther, Dena, and the wonderful technicians, Maria, Jose, Magda, Eva, Shafayat. I really couldn't do this without all of you guys. I want to thank the Neuroscience Graduate Program and my dissertation committee, all of our collaborators that we're working with, and Oliver, who I absolutely and utterly abuse at the Flow Core. And with that, I'll take any questions if anybody has anything.
Maureen, there is a question in the chat for you.
Oh, is there? Oh, there were 3. I'm so sorry. I'm not very great at looking at the chat. Let's see.
Regarding the in vivo studies using TNFR2 inhibitors, do they cross the blood-brain barrier?
Yes.
Dr. Dietrich.
Yes. So I've actually seen that in specific regard to these 2 inhibitors, they have been shown to cross the blood-brain barrier. And they are molecularly small enough to succeed in their crossing. So I should expect to see no issues with crossing of the blood-brain barrier with these 2 different inhibitors. Great question.
Well, I'll just say that it was a very nice presentation and very clear, and it looks like there's a lot of work to do, and you're exactly right. We have a lot of people around you that can help you with different types of techniques, and I think the more techniques you try to throw at this particular problem, and I'm very excited that Roberta and Dan have finally realized that moving from one part of the neural axis to the brain is kind of interesting in terms of targeting the TNF pattern. So look forward to seeing your progress and results, and thank you very much for a great presentation.
Oh, thank you so much. I'm also very excited. It really helps when you're really passionate about your research, and this is as cool as it gets for me, I think, personally. So thank you for your comments.